So when you're looking at structures, there are a few groups of elements, a few groups of bonded elements that help define the function of a molecule. So if you think of a car and how a car works, right? Um, so if you think of a car and you think, well, okay, the car has certain components that you absolutely are going to need to take into consideration if you want to call it a car. And certainly, I think, when you think about these things called cars, you would have to think of specifically wheels, right? Or tires. And tires have to be round. So you can take tires and you can put them on a car, or you can take tires and you put them on a truck. You can take tires and put them on all kinds of different vehicles, a motorcycle, for instance. You might think of taking maybe a radiator, you know, uh, uh, or a grill or a light. And each of these is going to have a different function and they have a different structure. So when you look at molecules, you also have these different components that have, like tires around and they allow the wheel to turn. And lights are electrical and they glow in the dark. And, an exhaust pipe is made of metal and can take a lot of heat and allows exhaust fumes to exit the engine. Those are groups that you put them together and you get a car. Well, here's some groups of elements, right? That when they're put on something, they give you certain qualities that allow you then to create something new, to create something with specific qualities. So now let's take a look at some of these. We've already talked about one group, the OH group. OH, also known as hydroxyl. It's an oxygen. Let me be clear on what this is. This is an oxygen with 8 protons, so there's 8 electrons. So it has, with 8 protons in its center, has an outer shell of 6 electrons, 6 negative charges, and allows this oxygen to make 2 bonds because it needs 8. It has 6, it needs 8, so it needs to make 2 bonds in order to share these extra electrons. And when it bonds with another atom called hydrogen, which has 1 electron in its outer shell, and it needs only 1 other electron to form its full outer shell. It forms this bond, this sharing of electrons, so that when you share, you'll note that the electrons belong to both of the atoms. Anytime you share, the thing that you're sharing belongs to both of you. So that's 2, 3, 4, 5, 6, 7 electrons. Oxygen needs 8. Hydrogen is sharing 2 electrons, so it's fine, it's happy. But oxygen needs 8. If you counted the number of protons and the number of electrons, you'd find out that this OH group, it needs to have 1 more. So it's going to be bonded to something else. Now, if you bonded it to hydrogen, well, then you'd have water. If you bonded it, right, H2O, and water would look something, and we've drawn it multiple times, it would look something like this. If you connected that OH group, OH, to a carbon, the way we've done it before, we've done this before, to a carbon that's also double bonded to an oxygen, that's then bonded to center carbon, that's bonded to a hydrogen, that's bonded to some variable R group, this bonded to a nitrogen, that's bonded to two hydrogens, each one filling out its octet, we would call that an amino acid. But this hydroxyl group shares a quality. This hydroxyl group, you'll notice this is a hydroxyl group, or I could have, highlighted the other H as well if you like. You'll find these groups in different molecules, but their groups have similar properties. Okay, so there you go. That's called a hydroxyl group. All right. OH groups are found in alcohols. So this is ethanol. The alcohol that you have, a lot of people have rubbing alcohol, is called ethanol. If it's 100% or 90+% alcohol, ethanol, and they call it rubbing alcohol. There's smaller percentages of this alcohol in alcoholic drinks. This is the alcohol that people drink. You do not want to drink rubbing alcohol. Likely it will blind you, it will do nerve damage to you, and it will do all kinds of bad things to you. Unfortunately, drinking alcohol can lead to alcoholism, and so people are addicted to alcohol, and that some people get so bad with their alcoholism that they'll actually drink rubbing alcohol as a desperate need to get their fix, and they end up killing themselves or doing serious damage to their bodies. But all alcohols end in this OL group, OL, end in OL. Now these suffixes and prefixes are going to be important, so when you're reading, you understand what you're looking at. Alcohol ends in OL. So any kind of ethanol and propanol and isobutanol or whatever it is going to be an OL, is going to have an OH in it. A bunch of carbons and hydrogens with an OH. This is called a hydroxyl group. All right. A carbonyl group is, if you look at a carbonyl group is a double bonded carbon that's connected to two other things. Anytime you have a double bonded carbon in the middle of a chain of stuff, we call it a carbonyl group. A lot like a carboxyl. Please notice that a carboxyl group has the carbonyl group in it. It just also has a hydroxide. So if you put these two together, you get a carboxyl group. Well, that's the acid part in the amino acid. That's the acid part in the amino acid. So here you have the carbonyl with the hydroxyl together, you make a carboxyl or carboxylic acid. All right. They call it an acid because this hydrogen can leave and become H plus, and that becomes then this end. If this is an H plus, increases the acidity of the solution it's in, because pH or acidity, how acid something is, pH is the negative log of the hydrogen ion concentration, is equal to anywhere, it's a range, right? So pH is a range that can go anywhere from neutral at 7 to acid when it's very low, let's say 1, so you can technically go to 0, but anywhere from, let's just go ahead and put 0 to 14. So for 0, you could have a very concentrated hydrochloric acid or sulfuric acid. For 14, you could have, so this would be like a very concentrated hydrochloric acid, just as an example. That would be a pH of 0. And something like a sodium hydroxide, very concentrated sodium hydroxide, could have a pH of 14. All right, let's just say that the acid is red for all intents and is red for all intents and purposes, and then the base is green for whatever reason. Okay, so pH of 14 would be a base, and a pH of 0 would be an acid. And everything on that side, everything from 7 to 0 is an acid. It releases that hydrogen easily, so it's an acid. And what that means is, when we talk about that, we're talking about is hydrogen ions, hydrogens without electrons. So the hydrogen is released from the group. The hydrogen was bonded to something, but the thing was so electronegative, and under the right conditions, the hydrogen is released and the electrons are kept by the thing that has a lot of strength, a lot of electronegativity. This is a 0, by the way, not an O. So in this group here, oxygen has a high electronegativity. It's very strong. Hydrogen, it'll hold on to its electrons. Under the right conditions, this hydrogen will be released without an electron. So it will form a hydrogen ion. And a bunch of those hydrogen ions in the solution will make that solution an acid. That's what it means to have an acid. If that material is, if the substance is a base, so sodium hydroxide, for instance, where it takes up the hydrogen, releases OH is another definition. So high OH, low free hydrogen ions. OH is would be OH by itself. That's where hydroxodium hydroxide, where the hydroxyl group, it gains the electron because of the oxygen, keeps the electron. And sodium has an Na plus, loses the electron. The OH becomes negative because the oxygen keeps the electron. The H stays bonded. Remember, oxygen needs 2 bonds, so there's an extra electron. It kept the electron, left the sodium by itself. The fact that this is in high concentration, these square brackets mean concentration, we call it a base. So the pH scale runs from 0 to 14, and that's normally how things work. There are some exceptions. If we look at some exceptions, the most acidic stuff is this fluoroantimonic acid, HSBF6. It's a super acid. It's like 10 to the 14th, 14 orders of magnitude stronger than something with a pH of 0. And it's incredibly corrosive. It's made in a laboratory, obviously. You're not going to find it in nature, at least as far as I know. And it's literally the strongest acid that I know of, that I could find anyways. Again, I'm not a chemist, so. And the most basic is an organometallic superbase called N-butyllithium or sodium amide. It's the strongest of the bases. They're so acidic and so basic that they're off the scale. You can't even compare them to normal acids and bases. Well, thank goodness we don't have to worry about any of that, because what we are only worried about what we find in living things. Living things don't deal with that. So what we deal with is anywhere from 0 to 14. That's the only thing you're going to have to worry about. But just know that there are these like super acids and super bases used for whatever purposes that aren't found in nature. And so that's why this is called an amino acid, because it has this group, this hydroxyl group, that under the right conditions releases the hydrogen. It becomes a hydrogen ion. It keeps the negative. And if you have enough of these, they release a lot of these hydrogen ions. We call that, it turns the solution acidic. So this carboxyl group is sometimes called a carboxylic acid or organic acid when it's found in a solution. Acetic acid is an example of that. This hydrogen's released. Acetic acid, you know it as vinegar. It smells strong, but it's acidic. That's why we use it we soak beef in it, because not only does it taste good, but that vinegar softens the meat, because it starts to react with the meat because it's acidic, and it makes the meat soft. So acid, and we do a lot of this in all kinds of cooking. If you know kimchi, we can do that with the same thing with cabbage. Kimchi is cabbage that's fermented, and it makes this acid, acetic acid. I think it's acetic acid. In any case, it becomes acidic, and it tastes very sour. Acids taste sour, and kimchi is one of my favorite go-to dishes with rice, to eat with rice. And it's fermented cabbage. It's cabbage that has acid in it, and it softens it up and makes it very easy to digest. In its ionized form, this carboxylic acid, again, the oxygen keeps the electrons. It becomes negative. It releases the hydrogen ion. Notice that the group stays, the carboxyl group, or the OH, the carboxyl group is still together. It just lost the hydrogen, becomes negative. It's called a carboxylate ion, but you don't need to know all that. The bottom line is that's why we call this an acid. It's found in this carboxyl group is found in acetic acids and amino acids. So it's common, just like the wheel on the car. All right, amino group. The amino group is just NH2, and here again, another drawing of what I drew earlier. The NH2 has a carboxylic acid group on this side. Again, releasing that hydrogen will make the solution acidic. On the other side is an amine group, and that's why it's called an amino acid. When it's ionized, it has a plus charge, so this actually acts like a base. It'll take on an extra. Remember, acids release hydrogen, bases accept hydrogens. This will accept the extra hydrogen. It'll become positively charged, so it's a base. So it becomes, the amine group becomes ionized. It becomes positively charged. The carboxyl group, when it becomes ionized, has a negative charge. So, and they'll become positively charged when there's a lot of hydrogens, and they'll become negatively charged when there's not a lot of hydrogens. So the environment helps determine whether it's the amine side takes on an extra hydrogen or the carboxyl group releases a hydrogen. So this is an amino acid. Sulfhydryl group, SH. This is very important for helping bend or fold proteins like your hair. So the sulfhydryl groups will cross-link. They'll form these, they'll cross-link. And what that means is that they'll make a disulfide bridge. So if you have a protein, I'm just going to draw proteins like this, and you have another protein, and maybe it's like this. And why did it bend? Because it had this SH group here and this SH group here. They lost the H's, but what they'll do is they'll bond to each other. So it bends the molecule that it's part of. All right, so this sulfhydryl group, sulfur, and S is sulfur, and H hydrogen sulfhydryl group is part of the reason that people have curly hair, right? They have a lot of these sulfhydryl groups, so there's a lot of these a lot of these disulfide bridges being made, and that helps bend the protein, make your hair curly. A sharing of electrons. Now, when you put product in or you heat it, you can break those sulfur bridges, those disulfide bridges, and then you wrap it around a curler, or you keep it straight and then you let it cool off, and then the sulfur, or you add a product that binds to the sulfur so it doesn't allow them to make the disulfide bridges. Then you call that hair straightener, or that's how you use an iron to break those bonds and then let them set as straight, in a straight form, etc. And then when you get it wet, when you add water to it, all bets are off and it goes back to its natural shape. And we'll talk about natural shapes in the next video. And then you have a phosphate group. Now, this is a very important group. It's going to be found in a lot of things like DNA and RNA. It's going to be found in, it's the backbone in DNA and RNA. It's going to be found in ATP, the energy for the cell. So it's a really, really important group. And it's just it's a phosphorus with 4 oxygens around it. It's a phosphorus with 4 oxygens around it. So phosphorus can make five bonds, not 4. So what's a phosphorus going to do? It's going to go ahead and double bond to an oxygen, because oxygen can make 2 bonds. That's great for this oxygen here, because now this oxygen is set. It has its 2 bonds. Of course, this oxygen is going to be bonded to something else. If it's bonded to, it can be bonded to a number of things. It can be bonded to ADP, then it'll become ATP, right? If it's bonded to that. If it's bonded to a ring of on a ribose, then we call it RNA, which we'll talk about when we talk about DNA and RNA. And if it's bonded to a deoxyribose, it'd be part of DNA. And this would be ATP. So these are the things that they would become if this phosphate is bonded to those structures. So phosphate's a it's a rock, it's bonded rock. It's a mineral there's a phosphate cycle in the earth, just like there's a sulfur cycle and a carbon cycle. So these are, sulfur and carbon come from minerals, from rock, and plants take it out of the soil. Both of those are out of the soil, and animals eat them. Otherwise, it's the only source of sulfur and phosphorus we have. Potatoes are very good at that. They're root vegetables, so they're very good to get some phosphate out of, especially if you eat the skin. So in any case, phosphate's incredibly important source. We get organic phosphates. This organic form of phosphate is found in plants originally, but there's inorganic phosphate that's a rock, right? All right. And then another important molecule that helps regulate DNA, helps turn genes on and off. It's involved in a lot of different functions. Is this group called the methyl group? It's carbon with 3 hydrogens attached to it. It can bond to a lot of different things. You can find a methyl group in a lot of ring structures. Here's an example. They give you an example here. CH3 on this ring structure, right? This is a component of, this is called cysteine. It's the C in ATGC. It's a single ring nitrogenous base. It's a base. And you'll find a methyl group there, as well as an amine group. There's an amine there. And there's a single bonded, double bonded oxygen, right? It's a carbonyl. So you have, let me do a different color so you can see that there's 3 groups here. You have a carbonyl, you have an amine group, and you have a methyl group. Those 3 together in a ring with some extra nitrogen and carbons form this C, the C in ATGC. You've heard for DNA, many of you have learned already, in DNA you have A, T, G, and C. Those are your bases. Those are different parts of the sequences you have to learn. And if you look at cysteine, the C, this is what the ring looks like. It has 3 groups, the amine group, the methyl group, and the carbonyl group. If you learn these groups, if you're an AP, you really should know these groups as a minimum, at least these groups. Then it'll help you understand the rest of these, the broader uses in different structures as we go along. So know these different groups. These are the groups you have to know. It's in your book on page 63 if you have the new book. In the old book, I'm not sure what page, but it's in there as well. The same materials found in all books. In the new book, it's figure 4.9. All right.